Hi, everybody, in person and on Zoom. Thanks for coming to today's IE² seminar. I'm Rebecca from the Panacea Lab, and I'm super excited to host Dr. Brian Spiesman today. And actually, we can say welcome back to Brian because he's from the Portland area and briefly attended UO at the start of his scientific journey. Then he went on to complete a bachelor's in biology at Portland State University, then went on to complete a master's degree in wildlife ecology and conservation at the University of Florida, followed by his PhD that he earned from Florida State University in biological science. Then he went on to do a postdoc in entomology at the University of Wisconsin-Madison. And currently he is an assistant professor in entomology at Kansas State University. And I first heard of Brian's really cool work last year at ESA, so I'm super excited to hear more about what he's up to. All right, thanks again for being here, Brian, and I'll let you take it away.
All right, thank you. Yeah, it's great to be here, virtually at least. All right, yeah, so I'm run the Lab for Pollinator Ecology at K-State, and I started here back in 2018, and since then we've been sort of, working on the realm of basic and theoretical ecology, applied ecology, and conservation. And lately we've been getting a little bit more into applied computer vision, mainly to facilitate a lot of the work that we do in the field. So I'm going to start off by talking a little bit about pollinator decline and some of its drivers, sort of as a backdrop, sort of as a motivating factor for some of the research we do. And then I'll talk about research on developing AI and computer vision tools for pollinator ecology and using crowdsourcing and crowdsourcing methods for conservation through community science and open data. And then talk a little bit about the outlook for AI-based methods in ecology and conservation. So pollinator decline, we have a lot of good information on pollinator decline from a few sort of well-studied species. For example, the monarch butterfly's overwintering area in Mexico is steadily declined since 1994. A lot of the bumblebees in North America, which were once common, are now in decline. So for example, taking a closer look at a couple of these, we have the western bumblebee, which is its occurrence probability across basically all of the states in its range is in steep decline. And the rusty patch bumblebee, Bombus affinis, is now listed as endangered, and we're seeing a steep decline in its range. It was once widespread throughout much of the Northeast and the Midwest, and now it's sort of relegated to a few locally common areas, but regionally it's undergone a very steep range contraction. So all of this is really driven by a lot of interconnected stressors that are kind of working together to cause this decline. So we have things like invasive species, habitat loss, pathogens, climate change, and pesticides. Habitat loss, for example, reduces the amount and access to food and nesting resources. Poor nutrition makes pollinators more susceptible to disease and pesticides, and then pesticides are more likely to be present where resources are limiting. And we can see how this all works together to reduce pollinators and their pollination services in some of the areas where we need them most. So in this study by Coe and colleagues, they looked at the dependency weighted crop area. And so you can see that here on the y-axis, and bee abundance, and areas where in pink are areas where we have crops that are highly dependent on pollinators, but also coincide in areas where bee abundances are low. And so we can see that this occurs in much of our important agricultural areas in the United States. So we have important research needs for pollinator conservation. And we know that bee abundance and diversity in many of these areas are in decline, but we don't have a good idea about which species are in decline and what are the real drivers of these. And we need to know this information so we can develop targeted conservation strategies. So for example, the IUCN Red List, so with over 20,000 bee species in the world, only about 167 or less than 1% have been sort of assessed by the people with IUCN. And 41%, almost half of these species are bumblebees. So it's really skewed toward the more charismatic species. So we need more species-specific data to map trends in both time and space. And we need new technology to help us gather these data on the large spatial scales and temporal scales that are important for pollinators. So they all have these many of them have these very large ranges. And so we need large-scale efforts to understand what's going on. And fortunately, we have the gain in popularity of things like citizen science and AI-assisted data collection to really help us out with this. And these can really work together to help us with these research needs. So as I said earlier, I've kind of got into computer vision and basically as a way to help out with this research bottleneck, bee identification. So we have thousands of species in North America and around the world, and they are highly challenging to identify. So for example, Lasioglossum, especially the subgenus Stylictus, has been called taxonomically challenging, morphologically monotonous, and even the despair of taxonomists. So this identification is very time consuming, it's costly, it requires expert knowledge. Lethal collection methods are usually required. We usually have to collect them in the field, bring them back to the lab, pin them, and then examine them under a microscope. So photo identification of a lot of these species is very difficult for humans, especially because a lot of these features, such as the thickness of this line on the mesoscutum, which is diagnostic for this in this particular example, a lot of these features are not well resolved in photos, or they're obscured. And so what we're trying to do is assess whether AI, artificial intelligence, and computer vision can help us remove this bottleneck for identification. So we're all, especially lately, even in like the last year, we've seen this increase in the availability and just the just in our culture, that artificial intelligence, but it's been around for for the last decade or more. It's just been a little bit more under the hood, but yeah, we're a lot of our cars now have at least some level of automation to them. We have AI on our phones, in our homes. It's sort of everywhere now. So we can use this in entomology and wildlife ecology and plant pathology to count pest insects. We can identify and locate pathogens, diseases in plants, and we can even use it to identify individuals, such as leopards here, where individuals can be identified by the patterns in their spots. And a lot of the AI and computer vision technology that has come out recently has been more geared toward sort of large wildlife, such as cheetahs and other things. But now we're starting to get a little bit more into being able to use and apply it in entomology and the ecology of small things like bees and other insects. So how does this work? Computers they view images as patterns of pixels, and so each pixel has a numeric value that can correspond to light or dark pattern or a particular color. So our computers read images and can analyze them as arrays of numbers. And so if we take an input image like this bee, we can run it through a convolutional neural network, which is up until recently, it's been sort of the state of the art. We're starting to get into newer technologies now, but with convolutional neural networks, we can pass the image through a series of filters, and the convolved outputs from these filters correspond to different features at different scales. And then when the model learns the features at these different scales, it can then sort of concatenate them and pass them through a connected network that can then classify the species based on the trained model. And so if, for example, if this was a facial recognition algorithm, the first layers in the network would correspond to things like lines and edges. The middle layers would be more recognizable features like eyes, noses, and ears, things like that. And then it would work up to eventually full faces. And the great thing about this, especially for identifying insects like bees, is that it only requires a set of pre-trained images as input for model training. We don't have to give it any additional feature input. The network learns its own feature set to make classifications. So for example, we don't have to tell it what a submarginal cell is. We don't have to tell it, that a particular pattern of coloration on an abdomen is associated with some species and not others. We just give it a list of, or a lot of images that are pre-identified that it can then figure out its own features that it then uses to make classifications. So the downside of this is that these neural networks are very data hungry, and we can see from this sort of array of images here, bees. If we're trying to identify bees from images, especially ones from the field, the bees are photographed in all kinds of different poses and in different contexts, different lighting, visiting different flowers. And then, on top of that, bees are, even within a species can be highly variable. So for example, Bombus flavifrons is highly variable in color, both across queens workers and males, but even within there's a lot of variation. And so this then means that we have to feed the training, we have to train the algorithm with a lot of different images so that it can learn how to classify species with all of this context associated with it. So along these lines, a lot of this variation, this intraspecific variation, is very spatially. So, for example, Bombus flavifrons has this more black and orange and yellow in the eastern part of its range, and as you move toward the west, it's mainly black and yellow. On top of that, so not only does it vary spatially, but what species it most represents also varies, or most resembles also varies spatially. So, for example, Bombus flavifrons in the western part of its range might more closely represent or resemble Melanopygus or Huntii compared to itself in a different part of its range. And in the western part of its range, it might more closely resemble a completely different set of species. So all of this makes it very difficult for the models to learn. But nevertheless, it seems to do a pretty good job, which I'll show here. So one of the ways that we can generate all this data, this image data that we need to train these models, is through these very important repositories of data. So, for example, the Global Biodiversity Information Facility, or GBIF, holds over, last count, it holds over 2 billion records, occurrence records across 85,000 different data sets. So not all of those have images associated with them, but a lot of them do. And the citizen science programs that contribute to GBIF are becoming more and more popular. So, for example, iNaturalist is growing almost exponentially in popularity. So, in 2021, there was at its peak over 250,000 records of insects were being contributed to iNaturalist on a weekly basis. And it's probably grown from there, since these data are a couple of years old now. So we are focusing on Bombus, at least to begin with, because that's where a lot of good data is available. So GBIF holds over close to 450,000 Bombus images across 235 different species. Many of these come from community contributed sources like iNaturalist. We also have different museums that contribute, and other sources. So even though we have all of this really great data, we still don't have enough to fully and accurately cover the the approximated 280 different Bombus species globally. So, for example, if in our models, if we wanted to include at least 1,000 images of each species, which is actually kind of on the low end, that would only give us images to include about 55 species in our model. And then if we arbitrarily decrease that sort of threshold to 150 images, that would only give us to about 108 species. So we need a lot more, even though we have all of this really great data out there, we're still well short of what we need to cover the full range of Bombus species globally, not to mention the other 200 or the other 20,000 bee species. So, and we can see where a lot of the problem comes from here is that bumblebees are highly diverse in North America and Europe, but the center of diversity is really in the Himalaya and China. And you can see on the in these GBIF records, this is areas where we have very few of a backbone to train the model, and this has resulted in an overall accuracy of over 93%, with a top 3 accuracy of over 99%. And what that means is that the correct identification is presented within the top 3 classifications over 99% of the time. And we can dig into this into these classification or into these training results a little bit more and look at them on sort of a species level. So each species has its own level of precision, and so that tends to vary by the number of images included in the algorithm for each species. So we have a lot of species that are really well covered. So Bombus impatiens is one of these top ones. It's one of the most commonly seen bee in North America. And then other species are less commonly seen, so they have fewer images associated with them. So as you sort of you decrease in the number of images included in the model, we see the potential to decline and have a decline in precision. But even for species that have very few images, there's a lot of variation in precisions, and this is, probably a lot of this is due to that variation, that intraspecific variation. So things like Bombus fraternus, which has relatively little intraspecific variation, has a pretty high level of precision, even though it was trained on very few images. especially compared to Silvicola, which is a lot more variable, and so therefore its precision is lower. And then, there's also there's many reasons why images can be misclassified, and so a lot of it really comes down to the quality of the image. So if we have images where a lot of the B is obscured, it's blurry, things like that, those tend to result in greater misclassifications. We can also look to use this data to sort of examine where we need to focus new data collection. So if we want to achieve an accuracy or a precision, a species level precision of at least 90%, then it looks like we need about 7,500 images to achieve that. But some species don't require that many. So if we sort of focus our new data collection on the species in this quadrant, then that will probably be, sort of get us the most bang for the buck. However, these are, based on their rarity, their proximity to urban centers, things like that. Those are typically the hardest ones to get, but nevertheless, it gives us sort of a target. So these algorithms are online at bmachine.ai. So anybody can go there, upload an image. It's important to crop out the image we want to reduce all of or as much of the non-B pixels as possible, and sort of feed in this this cropped image into the algorithm, and we'll get a results with the top 3 predictions. And then you can click on these to do a little a web search. We're currently developing a mobile app for Android and iOS, and this is almost done. So we have a version ready to go for Android, where we have a couple of bugs left to work out in the iOS version, but it should be available here very soon. So this will be covering bumblebee species in the Americas, Europe, parts of Asia. And this will have a lot more functionality than our website does, which really only gives you that ID. What we're hoping for here is more of like a citizen science type app. So users will be able to get IDs, plot their sightings on a map, keep track of their sightings, see educational content, and then all of the sightings that users upload will be contributed to to an ever-growing data set that will be available for research. And so crowdsourced data sets are important for our understanding of trends across time and space that no single research study could really accomplish alone. And so that's really why we want to contribute the data from the bmachine mobile app to some of these crowdsourced data sets. They're really important for understanding these trends that we really couldn't get at if we didn't have thousands of people out there interested in bees and other organisms to help us collect these data. So for example, Saori and colleagues use data from GBIF to ask how climate change affects bumblebee diversity in North America and Europe. They found that greater frequency of unusually hot days drives local extinction. And the result here is most areas of North America and Europe have seen some kind of climate-driven decline in the number of bumblebee species within an area. In another study by Zatara and Azin, they found that even though the number of bee species, bee observations in GBIF has increased in the last few years, the total number of bee species found each year has actually declined. So we're seeing more effort put out there, and even with more effort, we're seeing a decline in the number of different species that we can that's being seen. And so I really rely on these kinds of data, but they don't come without their without their downsides. So a lot of these data they're sparse, they're opportunistically collected. and they're usually collected near population centers. And all of these kinds of, or all of these, features of crowdsourced data can introduce bias into the data set. So another problem is the general public varies in their ability to identify difficult taxa. Luckily, we have a lot of expert taxonomists who are donating their time to verify a lot of these observations that are being contributed to places like iNaturalist. However, they're often not able to keep up with this ever-growing influx of observations. And so that's why we are looking to other data sources to sort of complement and extends the data that we can generate online. So one really great source of data is specimens from museum collections and research collections. So what we are doing in my lab, and I'm partnering with some folks at the USGS at the B Lab in Logan to digitize some of these specimens in a way that a lot of the sort of large scale digitization programs are not really sort of a different way than it's being done by these large scale programs. So for us, we really need a sort of a high throughput, relatively low resolution set of images. So we're more interested in gathering a lot of images really fast. And so we can do this with just a microscope that we have in the lab and a cheap microscope camera. So a lot of the current training or computer vision algorithms that we use to train our classifiers with don't require high resolution images. So we can take relatively low resolution images, take a pen specimen, photograph it in a number of different angles, and do this all relatively quickly and go on to the next one. And so we can do this with with whole bees. We can do it with bee wings. And so we developed a small data set to start examining how well these kinds of images work for identifying, especially these difficult bee species like this Dialictus. So with so we looked at 20 species across 6 genera and a pretty small data set of only 8,000 images, and we achieved an accuracy of close to 95%. And so we were very pleased with this because a lot of these bees are difficult even for the pros. They have a hard time with a lot of these species unless they they have to get them underneath the microscope and have a lot of training in order to really be able to provide good identifications for these. And so our training data set was identified by experts, and so we're very confident in our initial identifications going in, and very pleased with our results here. So we can take a confusion matrix like this, and which shows the predicted species versus the true species that we know, and so we can divide this into divide this between the Dialictus here in this group and the other genera here, which performed very well. There was no errors in the test set. We saw a few more errors among the Dialictus, which is which is to be expected. They're a lot more difficult to identify than some of these other species. So this tells us that we probably need a few more images to increase the accuracy of our classification model for Dialictus. So with this, we can sort of examine which species we need to target for more images. So what we want to see here is along the diagonal is 100% of the test images being correctly predicted as the true species. So for example, Lasioglossum admirandum had a precision of 71%, and it was mainly confused with Lasioglossum hitchensii. So looking at our B-wing data set, we also collected sort of an even smaller data set from these B-wing images. Again, we had 20 species, which weren't necessarily the same as from the other data set across 5 genera. And just from a small data set of 120 images, we achieved an accuracy of over 95%. And this is kind of this one I think is especially surprising to us, because even with such a small data set, we were able to achieve a high accuracy on using only a wing and we have many of these very difficult dialectics, right? So even the experts would have a very difficult time, and I don't know any experts really that would be able to identify to the species level individuals based on the wing alone. They usually require they need to look at other features on the whole specimen in order to make the classification. So this suggests that the algorithm is able to come up with its own unique feature set that includes features of the images that us as humans aren't able to visualize, or just haven't picked up on yet. So our next steps here are, we want to expand B-Machine to other taxa. So I'm currently in the process of training a genus level classifier that will be able to classify, in addition to those 108 Bombus species, we'll also be able to identify 65 common genera of bees, but also include a couple, a few, highly common species like Apis mellifera, honeybees, and eastern carpenter bees. So with all of this, we have 190 taxa trained on over 1 million images, and our initial training has come up with an accuracy of about 93%. And so this was just sort of a quick pass through it. So once we refine this, I expect to get that number up a little bit more. So this will be deployed on the B-Machine website, hopefully later this spring, maybe early summer, and then also included on the mobile app when that's available. And so we have, many uses for computer vision. And so one of them is basically, we want to be able to use it to collect data, but we can also, I use it to, to process images. So we can take, videos of this, things like this male bumblebee, and use object detection to, to pull out frames of, to pull out individual frames from the video that we can then use for training new algorithms. We want to be able to improve and augment our vision models with available data. So it's going to be hard to collect new data. That's one of our limiting factors. But with available data, there's a lot of things we can do to improve our models. So one thing is to basically find out where are these AI models looking. This can help us take better photos. It can help us determine what features are important for classification, and which photos in our data set are poorly represented by those features. So we could, if we know what photos will be most informative for training, we can reduce our training data set, which will make training a lot faster. And we can also potentially help taxonomists find new features to separate species. Another way to sort of augment our training process is by, instead of feeding in the the sort of a square crop, we can sort of feed in these sort of detailed cropped images that includes only the bee and reduces that background context, so we can potentially gain better inference that way. We can also use few-shot learning, and this is good for if we have species that are very poorly represented in the data set, or maybe even not represented at all. We can use textual descriptions instead of images, and that will allow us to sort of flag images that have species that aren't represented in our data set. And we can also use generative adversarial networks. And so, a lot of us have seen this, especially recently. So these are neural networks that basically they're generating deep fakes. So this is a deep fake of a bee. So this bee doesn't exist and never did. It was generated from random noise. And what these models do is it's a pair of models, competing neural networks. One is aimed at fooling the other one, and the other one is aimed at its goal is to not be fooled. So it's kind of this back and forth. It's sort of this coevolution of these two models that make better and better images that are harder and harder to fool anybody. So this is an image that of a I think it's a Bombus bohemicus, I think, if I remember right. And so the goal here is to be able to use these data for augmenting data sets with poorly represented species. Another thing which I think will have the most immediate impact is incorporating location context sort of directly into the neural network. So if we have things like GPS coordinates, elevation, habitat, what flower species a bee was visiting, we can sort of extract some of these contact features and then incorporate that directly into the neural network, and paired up with the visual feature set, we have this contextual features that can then be concatenated and incorporated directly into the classification model. So this is sort of an active area of our research right now. It's just to incorporate location context. So also, we want to the goal here is not to just incorporate, or not to just use these algorithms for identifying species, but we want to automate that process both in the lab and out in the field. So we can take our algorithms, our trained algorithms, deploy them on these edge devices like a Raspberry Pi, pair them up with a camera, and then use that to automate the process of counting bees, identifying bees, and even even sort of monitoring their behavior. So one of the things we're focused on in our lab is understanding sort of the community ecology of bees. What makes some areas more diverse than others? How does flora availability and foraging strategy factor all into this? And so one of the things we found previously is that the pollinators they're highly flexible in their foraging strategies, and so this is something that's been seen widely by a lot of people. And so they like to divide up their resources into to these sort of smaller modules, which can reduce interspecific competition. And for conservation, what this can tell us is that because this sort of niche partitioning really helps increase the persistence of species in a community, we know that we need to plant a diversity of floral species rather than a few abundant species to provide food resources for bees. And so this comes from analyzing interaction networks between plants and pollinators. And anybody who's done this before knows how time-consuming this can be, how difficult it can be to get good sample sizes. And so this is really what we want to, try to help automate this process using cameras out in the field. So my graduate student Caleb Sowers has been working on this problem and making some pretty good progress on this front in deploying our algorithms to automate the process of quantifying plant-pollinator interaction networks. I'm also working with James Krall at the University of Wisconsin-Madison, and he's taken this process a little bit further with the hardware and has devised this strategy to sort of network a whole series of cameras. So he has about 60 cameras that he's networked together here in a meadow next to a sort of a natural area, and he uses he networks them so that he can get away with fewer GPUs, and therefore greatly reducing the cost of this kind of automation. He uses cheap cameras. He uses sort of cheap Raspberry Pi computers to process his data. So in this setup, he had 60 cameras that he deployed, and he took time-lapse photos with about a photo per minute for each camera. Deployed it for 6 weeks. Looked at 17 different flower species, and resampled 3 times weekly within this grid, and eventually wound up with around 4.5 million images. And so, this is something that you don't need computer vision to generate all this data, but you definitely need computer vision to process the data once you have it. So once you bring it back to the lab. So what I'm working on with James is deploying our algorithms, onto his hardware setup so that we can process all of these images without going crazy. So we took our object detection algorithm. So this was the input image. We can use our algorithm to automatically locate the bee in the image, then crop it out, and then run it through our classifier to determine the species. And so this is really great because we can get very detailed, very high resolution, temporally high resolution data in this area with relatively little effort. So here's a representation of the 60 cameras. The different colors represent different flower species that each camera was monitoring over the time that they were deployed. We can zoom in on, so here's 2 days within this time frame, and we can zoom in a little bit further, and so if we take this and we sort of take this moving window, and we can get very detailed, very high resolution information on visitation rates. From this, we know what species each pollinator was visiting, so that we can get things like specialization. And then, yeah, so specialization, so it's basically the evenness of visits to different flower species. So this one would be a relative generalist that visits each species equally, on up to a specialist where it visits one species with much higher preference over the other. And so we can see how these, not only how visitation changes at these high resolution temporal frequencies, but also how other ecological factors change, like specialization. We have very high resolution data on how specialization changes, not only throughout the season, but on a daily basis. I mean, so we can incorporate temperature, other weather factors in here to see how things like daily fluctuations in temperature can change dietary specialization. And I'm not going to go over the details of his results here, but the sort of the upshot here is that they found that short-term variation or short-term temperature fluctuations can affect both activity and dietary specialization. And this is additionally an effect of daily weather variation, but also the the effect of that on the strength of intraspecific competition. So just to kind of wind up here, I'm also working with other researchers here at K-State, so Dr. Brian McCornack and Dr. Yvonne Villalba. We can use these same kinds of algorithms and train them with different image data sets to tackle different problems in entomology and agriculture. So for example, Yvonne here for his PhD developed a series of different ways to quantify aphids on leaves using computer vision. So one of the ways is just use object detection, and as you can kind of see here, there's the aphids are so small in the image, yet the algorithm is able to detect most of with a pretty good accuracy, detect most of the aphids on here, and then give you a count of the aphids, average it over a number of different leaves. And so this is important for things like crop scouting, where there's a threshold in pest density where if it's above the threshold, it's recommended that crops be sprayed. If it's below the threshold, it's recommended that they agriculturists not spray yet. So if we deploy these algorithms on in automated systems like these robots that sort of crawl through the crops to determine where sort of targeted areas where it's most beneficial to spray at certain times of the growing season, then we can really reduce the amount of pesticides that we need to apply to crops. And so, yeah, just to wrap up here, so we're finding that, we think computer vision will be transformative for ecology and entomology. And a lot of these algorithms, these computer vision models work very well, but we're still, very limited by high quality data to train them. And hardware and software are being developed now. They've also been, sort of previously have been limiting, but we're starting to develop the hardware where we can actually get these algorithms into the hands of users so that they can be used for good. And so with that, I'd like to thank some graduate students here in my lab, Caleb Sowers, Maggie Anderson, and Jessica Butters, and then collaborators at University of Wisconsin, Claudia Breton, James Kral, and then other collaborators here at K-State, Bill Hsu and Brian McCornack and Dr. Tanya Kim. And if I have time, I hopefully can answer some questions.
Okay, first question.
Hey, thanks for the great talk. I was wondering, so you have a limitation of the number of pictures you have for training your model. And you talked about a little bit like how to crop the pictures to have more like a picture that is centered on the organisms. And I was wondering, there is also like another paradigm, which would be like data augmenting your data. So basically just like giving some kind of like filters where it's like more light, less light, like make them maybe low quality because it's something that you don't really control because you use a lot of like citizens data. So I was wondering, if instead of like just making it more specific on the organisms, you could just like data augment with a lot of filters, and maybe it will affect, of course, like the training time, right? It will be longer, but then maybe you will be able to like have more pictures to use.
Yeah. So I think if I understand your question right, we do use we incorporate that into the training process. So we call it data augmentation. So each image in our training set, we can sort of skew it, we can rotate it, we can crop it in different ways, we can we can apply different kinds of filters to it, and so that each round, each iteration of the training process, the algorithm sees a completely different data set, so it sort of artificially inflates the number of training images that we have. And that really helps out a lot. We see significant increases in accuracy when we use that process over without it. So, but the other part of that is we're also using this some of those attention diagrams to sort of like what parts of the image are really important, and so if we can feed into the algorithm, if we can feed it only the important, we can basically weed out the non-important stuff and then feed into it only the important features, and that could also help to increase the quality of the prediction.
Okay, great. Yeah, thanks for the answer.
Okay, next question.
Hi, thank you for the talk. It was great. When I was thinking about how taxonomists classify like an animal that they see, one of the things that they use first is what you mentioned already, like spatial context, or even like the flower context and stuff like that, which you talked about. But the other thing is, like, sometimes we may not be able to classify the species itself, but we will have some knowledge about the phylogeny. And so I was wondering if you've thought about doing hierarchical classification.
Yeah, that's a good that's something that we want to include. So we haven't done that yet, just because we've been sort of focusing on these bumblebees, like a particular set of bumblebees. But once we start branching out into other organisms, other kinds of bees and bee mimics, things like hoverflies and things like that often get confused for bees by some of the general public. We want to be able to include, that sort of hierarchical classification where maybe the model can get it to the family level or the genus level, but it can't quite get the species level. I think that'll be really good for it'll be good for what we're doing, but I think it'll also give maybe a little bit more confidence to the people that are using it, because they might be sort of suspect of a species level identification when they're not really sure, and it's hard for them to sort of verify. But I think that's a great idea.
A follow-up on this is, did you look at the errors? Does it make errors consistent with phylogenetic distance, like at the top tree classifications?
So no. Do you mean that things that are more related are going to be either better predicted or not as well predicted?
Correct.
Yeah, no, that's a good idea. We haven't looked at that, and it's the overriding thing so far is just the number of images, but then there are certainly groups that look a lot alike and that are very closely related, and so they may, just because they're more closely related, they make it they may be more often confused. But it'd be good to see if there's certain groups that are just poor performing and other groups that are well performing, regardless of the number of images that is included in the training.
Okay, another question.
Yeah, thanks. That was great. I have two questions. One is how your like sort of specialized bee-focused accuracy compares to a generalist method like iNaturalist's predictions. And the other one was if sometimes people might take more than one photo of the same insect, and is that information you can use?
Yeah. So okay. Yeah. So we're focusing more on a subset of taxa, especially compared to iNaturalist, which is everything. So our models do a lot better for bees and for well, it does better for bumblebees than iNaturalist does, and
Do
partly
you know how much better?
I haven't compared it because I don't know. I probably have to assess their their full model and sort of compare it directly with different with the test set, and I don't know if they're. I think only part of their model is available publicly. So it'd be but I think that would be definitely interesting to know. I just sort of anecdotally, I know that the sort of recommendations that iNaturalist gives, our model performs much better than that. But no, I haven't done sort of a rigorous test using a sort of a standard test data set or anything like that. And then sorry, what was the other question?
That sometimes people take more than one photo of the same organism, and is that, can you use that somehow?
Yeah, that's an interesting question. So one of the sort of the easiest way to do that is to process each image individually and then take like a maybe weight them somehow and sort of take the average of their predictions and then sort of give a combined prediction, or like maybe even something more simple like a vote counting or something like that. But it'd be interesting to, we haven't yet explored any way to sort of combine that into, combine two images into one, maybe use one as like a prior or something like that, or in some way. But yeah, we haven't explored that level yet.
Another question.
Hi, yeah, I am curious about the application of this kind of processing to specimen that maybe their taxonomy is a little bit more messy. So like, I think bumblebees are a great way to start because they are pretty well documented compared to things like Lasioglossum and like Nomata, that maybe a lot of the species are still undescribed. So I'm kind of curious how that works, like if you're essentially feeding the program this like list of species, but then maybe it encounters one that isn't on your list, like how will that kind of be resolved? And how do you see that working for, again, taxonomic groups that are not as organized like bumblebees?
Yeah, so this is one of the problems of this sort of technology, and this is why we really rely on taxonomists. And this technology is not going to replace taxonomists anytime soon. So we need the taxonomies resolved before we can really deploy models like this. So if you it's only good, the algorithms are only as good as the information that you give them. So if and it only knows what it knows. So I mean, I could feed a picture of a honeybee into our algorithm, and it would classify it as some kind of bumblebee. So it doesn't know, it just doesn't know honeybees. So they won't be able to know species that haven't been described if they haven't been trained on that. However, one of the things I mentioned was using the sort of zero-shot learning as a tool to discover sort of abnormalities or things that are out of the range of the normal range of variability of species. So if we see potentially new species that's not included in our model, like a honeybee, then it would show variation in feature set that is sort of out of the range of the species that are in there, and we could flag it as something that's potentially new. And that way would be a good way to sort of take care of the species that aren't included in the model in a way that provides some information to the users that it's okay, it's probably not one of the list of included species, it's probably something different. And then you could sort of take a closer look at that on your own.
Shall we thank Brian again? Yeah, thanks a lot. That was very informative. Too bad you're not here. Hope to see you sometime. Thanks.
Thanks, everybody.
Here is okay. Yeah.